Welcome to Click for IB Biology. We're in C3.2. We're studying the defense against disease. And in this particular video, we're going to be looking at immunological memory and how that links to the process of vaccination, the use of vaccines. There's a PDF of this presentation. It's linked into the description down below the video. And there you'll also find a link to 10 short answer questions which with answers provided, and you can use those to check your understanding of the content of this particular video, this particular part of the course. So immunological memory and vaccines. So in the previous two videos on the humoral and the cytotoxic response, the first encounter with a pathogen, so when we first come across a pathogen for the first time, it's never been in your body before, the first time it gets entry past the barriers to infection, past the clotting, past the innate system, this is called, what is the first encounter is called the primary response. And you'll recall from the mechanisms that I've shown you that we produce B memory cells, and those are in high frequency. And we produce antibodies, which are in high concentrations, shall we say. And we, from the cytotoxic side of the system, we also have cytotoxic memory cells, and those are in high frequency as well. Remember that previously in the primary response, the frequency was much lower. Let's take a look at the primary response and the secondary response. This gives us a good framework on which to understand vaccination. When we are first infected with a pathogen, this of course is the primary response. And although we have looked at barriers to infection and clotting and the innate system, we're concerned here largely about the adaptive system and the humoral system and the cytotoxic system. So after a period of time in which the pathogen is circulating within our plasma, our blood system, and within our tissues, that time, this time delay which we see in the diagram here, in which the disease is incubating, during this time, you would start to show the symptoms of that particular disease. And that effectively shows the damage that that disease is doing you. Now, your immune system in this time is selecting the correct B cell. It's looking for and selecting the correct T cell. So finding those low frequency cells within the lymph nodes of the two kinds. And also that's the first step. And the second step, you'll recall, is the step in which those cells will actually form the clone. Now, this takes time, and this is the time indicated here on this infection chart here. The red line indicates antibody production, and of course this is a B cell response, and as the antibody production comes up, the concentration will reach a level, the antibody required for immunity, and now what's happening is the agglutination, opsonization of the pathogen, and it's engulfing by the innate system's phagocytes. So we're into recovery, and we can see that this reaches a peak. So by this stage, once the B cells and the T cells are responding, then you are in recovery. Now, for a primary response, the antibody level never gets particularly high. And after the infection and when we're in recovery, the antibody level will slowly decline over time, with a kind of curve that goes more like this, really. Now, interestingly, if one then encounters the same pathogen again, now it has to be exactly the same pathogen, so we are reinfected, and so we will call that secondary infection. Now, notice that with a secondary infection here, that the production of antibodies is greater, and the time taken to respond is far, far quicker. Now, this is because already present, we have B memory cells in high frequency, And so there's no time taken to actually hunt down the specific B memory cell. They're such a high frequency, they will be selected very, very quickly by the pathogen. And of course, the T memory cells as well will also be selected very, very quickly for any cytotoxic response. But remember, there's also antibodies present within the bloodstream anyway, which will assist you in not actually developing the symptoms and signs of the disease with a secondary response. So typically, where it's an identical pathogen, the secondary response produces such a high level of antibody, because there's many B cells, that you don't actually experience much or any of the symptoms of that particular disease. And then you will be going into recovery. Indeed, one may not actually feel ill at all. And so now we have this enhanced antibody level, which will take time to decline. We have got, once more, we've got larger numbers of B memory cells and T memory cells as well. Okay, so this brings us to the discussion on vaccination. Now, if you think of the names of the different vaccines which exist, coming to mind immediately are things like measles, mumps, rubella, cholera, typhus, and so forth. These diseases, smallpox, these diseases, of course, are characteristically known to be very, very dangerous. And the reason we want to produce a vaccine is that in this time period here, for a primary response, if a person is placed in the position of producing a primary response to these very dangerous diseases, then the time it takes to actually produce the B cells and the T cells takes too long, and the damage which is done in this period can either create permanent physical disabilities, or potentially it could actually be a fatal disease. So we wish to avoid the situation of coming into a primary response with something like measles, mumps, rubella, cholera, and smallpox. We don't want to encounter those in a primary response, because our bodies will probably not produce a response fast enough to avoid damage or even death. So is there a way in which we can prepare our bodies with memory cells of the B type and the T type and antibodies in the bloodstream without actually going through the dangerous encounter with the real living thing? And that's what vaccines do. So vaccination is to create a primary response without the danger of the pathogen causing damage. We know that the primary response is going to produce antibodies, B cells, and T cells, both high frequency in the plasma and particular memory cells. And they'll be located not just in the plasma, but also in our lymph nodes. And the way that we do this is to produce vaccines, so-called vaccines, which are composed of either completely dead pathogens. Now, the significance of a dead pathogen is it can't replicate and therefore cannot spread the disease within your system. So one could have a vaccine of a dead pathogen. For some of them, that doesn't work, so we have to do what's called attenuated pathogen. It's extremely weakened. It's not actually dead, but weakened so that it too cannot replicate. And then there's another more recent type of vaccine, which are those based on the molecules of DNA and RNA, where those DNA and RNA molecules code for the antigen of the pathogen. If we introduce these vaccines into our system, we will produce a primary response which will result in B lymphocyte memory, T lymphocyte memory, and antibody things, but we will not actually experience any disease. Should we then encounter these dangerous pathogens, we already have an immune system an adaptive immune system which is prepared and ready for a rapid secondary response in which we will not experience the disease or the damage of the disease or the symptoms of the disease. In other words, we have immunity from vaccination. A question which is often asked about this vaccination process is how many people need to be vaccinated to protect the entire population. And this is a concept called herd immunity. So in the diagram, you can see that these people here are susceptible and these people here are infected. And so where we have close contact between the infected and the susceptibles, there is rapid transmission of the disease through the population. But you can see here in the second diagram, where vaccination has occurred, that the people in green have been vaccinated. They have already gone through the primary response. And so whilst we have the infected individual here, perhaps infecting one or two people, there's a barrier of immunity around those individuals, preventing them from the transmission to the susceptible people. Now, the question might then be, what percentage of the population needs to be immunized in order to create this herd immunity? And that depends very much on the nature of the disease, on the virulence of the disease, how serious the disease is, how easily it's transmitted from one person to another, and the mechanism of transmission as well. So for instance, for some diseases such as measles, we require as high as 95% immunization in order to protect the susceptible individuals. One should note that not everyone is in a position to take a vaccine because of their immunocompetence. That is to say, some people are actually recovering from transplants or other autoimmune diseases or immunological conditions that vaccination is not appropriate. And so we vaccinate the rest of the population in order to protect these susceptible and vulnerable individuals. That is the principle of vaccination. There's a link now in the window in front of us to the next video. A chance to subscribe. Please do that. It's greatly appreciated. And there's the playlist for your convenience.